Hello students. So before I go on to today's lecture on the linkage and crossing over, I would first like to recap on the law of independent assortment. According to the law of independent assortment, the alleles of two or more genes get sorted into gametes independent of each other. The allele received for one gene does not influence the allele received for another gene. As we know, alleles are two or more different forms of a particular gene. And so, in Mendel's cross, in dihybrid cross, Mendel had crossed round yellow homozygote peas with that of wrinkled green homozygote peas. And here the genes are for the genes for the shape of the pea, and the next is the gene is the color of the pea. And so the different alleles are, one is round and the other is wrinkled, and for the other gene it is yellow and green. So when Mendel crossed the parental homozygotes in the first filial generation, he got all of them were round, all were round and yellow, and the green wrinkled were not present here. And so when he selfed these things in the F2 generation, as we know famously, he got them in the ratio of 9:3:3:1. So he got two new characters. That is, he got a yellow wrinkled and a green round for the first time. So in this case, there was an independent assortment of the gametes. And this phenomenon made him give this principle of independent assortment, that present of round in no way influences the color of the pea. It can be green or it can be yellow. So this was independent assortment. But this, in later studies, it was found that this was not true. Sweet pea had something different to say, and that was linkage. So this is a picture of a sweet pea. So linkage was between genes was first discovered in experiments with sweet peas. And this was a very famous experiment where it was a deviation from the 9:3:3:1 crosses. So with this, let us start the linkage and crossing over. So, Mendel's independent assortment holds true when the genes are located on different chromosomes. This is as we know today. And also we know from cytological studies that a phenomenon takes place in meiosis, and that is known as crossing over between non-sister chromatids in homologous chromosomes. As we know that each cell of our body contains a set received from your father and a set received from your mother. So for 23 from dad, 23 from mom. And so during meiosis in our gametes, a type of crossing over takes place between non-sister chromatids. One complement which had been received from your father and one had been received from your mother. And now you're making a new sort of complement. And so crossing over takes place and you have a sort of exchange of DNA between these non-sister chromatids. This you pass on to your children. So, independent assortment occurs when these particular alleles or these particular genes, for example, are located on different chromosomes. But when they are on the same chromosome, there is somewhat the gene which is located, for example, in the red arm, somehow has got a tendency not to leave another gene which is located just beside it. If a gene is located here, it would not allow somebody just beside it to be exchanged. So there is a sort of pull that no, don't go, stay with me. Imagine in a rubber band, it's very difficult. A unit from here is difficult to separate it from here. The farther they are away from each other, it is more easier for them to separate. But the closer they are together, there's the chance that they are transmitted together is much higher. If they are far away, then the chances are less that they are transmitted together. But when they are in the separate chromosome, then independent assortment starts to take place. So deviations from independent assortments was first observed by Bateson and Punnett in 1905. Trait in sweet pea that flower color can be either purple or red. Purple is dominant to red. And pollen shape also can be either oblong or it can be round. Oblong is dominant to round. So when such a thing was done by Bateson and Punnett, where he crossed purple long with that of red and round, in the F1 offspring, as expected, it was purple and long. Purple flowers, long pollen. When they were crossed with red flowers, round pollen, they got the F1 offspring, they got purple flowers, long pollen. And F2 crossing of this crossing with PL and small p capital L and small pl, all these things, they possess 4 different phenotypes. F1 offspring had purple flowers, long pollen, and after self-fertilization, they were found in this ratio. It's supposed to be found in this ratio, 9:3:3:1. But what was observed was that a ratio of 15.6:1:1.4:4.5. So there was a deviation in such a phenomenon. And this did not follow independent assortment. So for the first time, one could see a deviation from independent assortment. So in the F1 and F2 generation, the observed ratio is 15.6:1:1.4:4.5. The expected ratio 9:3:3:1 is not seen. The parental phenotypes were overrepresented. Instead of 9:1, you have 15.6:4.5. And non-parental phenotypes, instead of 3:3, they were underrepresented. This was evidence that the two genes do not assort independently. The nature of their physical linkage was not apparent to Bateson and Punnett at that time. The genes were physically linked on the same chromosome. So, linkage can be defined as the close association of genes or other DNA sequences on the same chromosomes. The closer two genes are to each other on the chromosome, the greater the probability that they will be inherited together. Like I mentioned, the closer they are, chance that in the next generation both of them will go together. Further they are, the chances are less. This is because when they are located in the same chromosome. This chromosome theory of inheritance was established by Sutton and Boveri, and some famous experiments done by T.H. Morgan in Drosophila established the phenomenon of linkage. So, let us look at this cross. Red, long, red, long. These are the dominant. And white, short, small r, small l. When they are crossed in the F1, you have this sort of a phenomenon taking place. A heterozygote is formed. And when meiosis takes place, there one place when there is no crossing over, when the parental phenotypes are taking place, parental phenotypes are coming, this is where no crossing over is taking place. But when crossing over takes place, you find these 4 types of gametes being formed. One is the parental capital R, capital L, and the parental small r, small l. And you have the recombinant chromosomes where you had capital R, small l, and small r, capital L. So, genes that are on the same chromosomes travel through meiosis together. However, alleles of chromosomally linked genes can be recombined by crossing over. So, recombination takes place, crossing over takes place. They are linked with each other when they are on the same chromosomes. So closer they are, chances they are inherited together, they go together. But the farther they are, chances are less, but still they can go together. So now we have phases of linkage. One is a coupling phase and one is a repulsion phase. So a coupling heterozygote has got something like this, where no recombination has place. Capital R, capital L on one chromosome, and on the other you have small r, small l on the other. This is a coupling heterozygous. And then you have the repulsion heterozygous, when you have capital R, small r, capital L. So next we go on to determining the linkage and frequency of recombination. So let's look at this cross again once more. Capital R, capital L, and small r, small l. It is crossed with small r, small l, small r, small l. So they are crossed with each other. So when they are crossed with each other, so what do we expect in the F2? We expect these things when crossing over. These four types of combination takes place. You have capital R, capital L, small r, small l on one side. You have the other small r, small l, and small r, small l. This phenotype. Then you have the recombinants coming, capital R, small l, and of course you have the other complement, small r, small l. And you have the other way round, the small r, capital L, and R and L small on the other chromosome. So when such a selfing has been done, you find the ratio. You find the parental ratio. In this particular, this type of cross is known as a test cross. What you find here, you find 920 parental forms are found in comparison to that of 80 of them in the recombinant form. This is just an example. It can be different for different genes. In this particular cross, you find it to be like this. You find red long 450 of them, and you have white short 470. So you have the parental classes are overrepresented too much. And you have, for example, 80 recombinants. The recombinants which are formed, they are less, 42 and 38 respectively. So now how do you calculate the frequency of recombination? So the frequency of recombination can be calculated as there are total of, say, 80 plus 920, that is 1,000 of these plants have been found, and you have the recombinants, you had only 80 recombinants, so out of the total number of plants which are found in the F2 generation, 80 of them were recombinants, and so the frequency of recombinants is 0.08. And this is also known as the distance between these two genes, R and L. So the key points from this lecture are: linkage between genes is detected as deviation from expectations based on Mendel's principle of independent assortment. And the frequency of recombination measures the intensity of linkage. In the absence of linkage, this frequency is 50%. For very tight linkage, it is close to 0, and intermediate in between. So thank you, and in the next class we will go for the next part of linkage and recombination, where we will be discussing about Drosophila 3-point crosses and the linkage and recombination, how it is used to map distances between genes. Thank you.